All right, so today's lecture is on mitosis in the cell cycle. If you want to know where it actually is in your book, it's chapter 12, if you want to do some review on that. But we're going to look at what is the purpose of doing mitosis? What is the purpose of doing cell division? Why do cells need to divide? If you're a single-celled organism, you want to do it to reproduce, to make more copies of yourself. But in a multicellular organism, the purpose of mitosis or cell division is to grow, develop, and change, and also to repair when you have some sort of, if you have a cut or if you have an injury. So back to living environment, there are two major types of reproduction. There's asexual reproduction, and remember that involves one parent. You have identical copies of cells that you are making, and that's barring any mutations that are caused by the actual copying process. Remember, we talked about that when we went over DNA replication. Sometimes mistakes happen. Usually it fixes them, sometimes it doesn't. And asexual reproduction also happens very quickly because you don't have to find a partner in which to reproduce with, so you can just reproduce yourself. Sexual reproduction, on the other hand, you need two parents for that. That involves the production of gametes or sex cells by the male and the female, and then those sex cells fuse together. And this process is important because it actually creates genetic diversity, and genetic diversity is very important for the survival of the species, which we are going to talk about when we get more into evolution. So this lecture is just going to focus on asexual reproduction, really, and specifically how eukaryotes do asexual reproduction through the process of mitosis. But before we get into that, let's talk about some differences again between prokaryotes and eukaryotes. I know we've talked about this several times, but prokaryotes, remember, have no nucleus and no membrane-bound organelles. So that'll make the process of cell division a little bit simpler than if you were a eukaryote, because they have a true nucleus and many membrane-bound organelles. So if you're going to copy a cell, you're going to have to copy all those organelles as well as make the nucleus disappear and copy all of the genetic material. These are different types of eukaryotes. We have humans, plants, diatoms, and a generic eukaryotic cell, as well as insects. But there are many different types of eukaryotes aside from this as well. All right, so some different examples of asexual reproduction before we get into mitosis, which is the third one on this list. Prokaryotic cells do what's called binary fission. It's a very simple form of asexual reproduction where the DNA is copied and the cell divides into two. Some protists and fungi do a process called budding, which is essentially like me, if I were to bud, like a head would grow off the side of me, and then eventually a little body would form, and then it would pop off and then run away. That is what budding is. So it's like I make a copy of myself from myself, usually multicellular organisms, with the exception of certain types of fungi. Those are single-celled, like yeasts. They do the process of budding. It's just kind of like an unequal splitting of the cytoplasm, and then that tinier bud will grow and form a new organism. But most eukaryotes divide by mitosis. So this is showing you binary fission. Notice the DNA is replicated through that same process we talked about when we went through DNA replication, right, with helicase and ligase and primase and all those things. It copies itself, and then the cell wall of the bacteria will split down the middle. It'll form a new cell wall between them, and eventually we'll create two separate cells. And this is showing over here actual binary fission using electron microscopes. So it's very simple. Here's showing you budding, and what I mean, notice this tiny part right here is budding off of the original eukaryotic cell, in this case yeast. And this is showing mitosis. This is going to be the process that we're going to be focusing on in a little bit. There's also this process called vegetative propagation, which is another type of asexual reproduction. And essentially what happens in this is instead of producing seeds and fertilizing those seeds, which is sexual reproduction in plants, a plant will send out a shoot or a root to the side and then just create a new individual directly from that. So every single one of these plants in this line is genetically identical to itself. And this exists and is very common throughout the animal kingdom. And we can see it in things like strawberries and even in Japanese knotweed, which is an invasive species that has come over from Japan that we have in this country now. So, all right. So before we talk about mitosis, we have to talk about the nucleus. The nucleus is an organelle, remember, that stores the genetic information. It's the control center, and we know it's the control center because it houses the DNA. And DNA is the recipe book for making proteins, and proteins do everything. So this organelle has two functions. It stores DNA, and it also coordinates the cell's activities by kind of housing that genetic material and allowing things in and out that will control genes and control genes being turned on and off and things like that. So if you think, if a cell wants to split in half, what are the types of things that it's going to want to do in order to make sure that the new half or both halves have what the original cell had? You're going to have to replicate the DNA because you need a new copy of the recipe book. You can't just split it in half. You'd lose half of the instructions for making proteins. You'd also have to copy all of the organelles. You'd have to increase the cytoplasm because it's going to grow larger. You don't want to be much smaller than you were originally. And you also want to make proteins that are necessary for division to happen. And those are all things that the cell will actually do before it goes through mitosis. So there are three main phases, if you count cytokinesis, two if you don't, of the cell cycle. Interphase is kind of its resting phase. That's when the cell spends most of its time, is during interphase. During this time, the DNA is replicated. It prepares itself to divide. But the key for that is it prepares to divide, and it also is kind of doing its thing. And it spends most of its time in this phase. Mitosis happens relatively quickly. We're going to talk about the different phases of that, and that's essentially where they take the DNA that has been replicated and splits it between the two cells. And then cytokinesis is the process of actually pinching the cells into two new cells physically. So you can see cells in interphase, and we're going to do a lab on this, but the cells in interphase, if you look at them at the nuclei of the cell, you can't tell that there's different chromosomes. They're not separate. Whereas when a cell is ready to divide, it starts to separate into individual chromosomes. It condenses. In this one, during interphase, the DNA is mostly in the form of chromatin, which is, remember, that jumbled up mess of DNA that's throughout the whole entire nucleus. It's not until it divides that they actually condense into chromosomes that we can see. Okay, so let's look at interphase, and then we're going to talk about how the cell actually controls this cycle, controls the process of mitosis in interphase. So interphase, as I said, is the resting phase. The cell is just doing its thing. It's making protein, it's digesting, it's living. Eventually, the cells will also have to do the things that are necessary to go into the mitotic process, which is duplicate chromosomes, duplicate organelles, grow a little bit. And we'll talk about each individual phase right now. So the interphase is actually split into three different phases, G1, S, and G2. G stands for gap, so gap 1, and then G2 is gap 2. The way I like to think of G2 is also growth, because in both the G1 and G2 phases, you have cellular growth and size and the production of proteins and things like that. So in G1, the important thing that you have to know about is that proteins are synthesized associated with mitosis, and it grows. Those proteins are called histones. And remember, histones are important for condensing the DNA and making it into chromosomes as opposed to just chromatin. There are histones in chromatin as well, but there are more histones that allow for more wrapping and more condensing once the chromosomes actually form. During S phase, that's also known as synthesis phase, you may want to write that in your notes, is when the DNA is replicated. So this is, during S phase is when DNA replication actually occurs. Again, the process where you take helicase and ligase and DNA polymerase and you make copies of the DNA. G2 is the third phase, which happens right before mitosis. In this phase, we have rapid cell growth because it's going to get ready to divide, and synthesis of proteins that are needed for mitosis, microtubule proteins, spindle proteins, and organelles are also replicated during this phase. So the cell is really getting ready to ramp up the fact that it's going to want to divide soon. So here's showing you the three phases. Notice G1, S, and G2. So in G1, we have one copy of the DNA. In G2, or during S, we copy the DNA. because that's the point of it, that's synthesis. Then G2, the cell prepares to divide, replicates the organelles, gets a little bit bigger. And then finally mitosis, which is a very small part of the cell cycle, is where that DNA is split in half and the cells are split into two. Again, notice how long interfaces G1, S, and G2 takes up most of the time of the cell cycle. Now, many cells will actually go into something called G0. That's when they're not actively dividing anymore. Nerve cells do this, muscle cells do this, other cells do this as well. But let's say there's an injury to some part of your body. The cells will then go from G0 back into G1 and then go into the cell cycle in order to make copies of themselves to repair that damage. So G0 is like a cell is just doing its thing, it's living, it's not even thinking about dividing. So make sure you understand G0. So how were they able to actually figure this out? It's kind of simple actually, but what they did was they added radioactive nucleotides to cells. And they wanted to see when the actual radioactive nucleotides were used. They found out that they were used during S phase, which is when DNA synthesis occurs. So this is a very simple experiment that they used to figure that out. And this is showing a nucleotide triphosphate with a radioactive label attached to it. So this cell cycle is actually controlled in very specific ways, because if the cell cycle goes out of control, do you know what happens? Cancer occurs. So the cell cycle needs a way to control itself so cells don't go haywire. Now, obviously, if we mess with those controls, that's when cancer kind of will take form, and we're going to talk about that a little bit later. It uses, or it controls this through things called checkpoints. These checkpoints occur several times during interphase and even during mitosis. So the purpose of the checkpoints is to monitor DNA replication. Did it happen properly? Are there mistakes that were made? Are there lots of mutations? If the DNA has not been properly replicated, the cell will not go to the next step. So these DNA damage checkpoints actually occur before the cell enters S phase. It's called the G1 checkpoint because it happens right before S phase, which is in G1. The DNA replication checkpoint, which is a G2 checkpoint, and then the M checkpoint, which makes sure that all of the chromosomes are lined up perfectly in the middle during my metaphase. So this is kind of showing you those checkpoints right here. We see the G1 checkpoint, the G2 checkpoint, and the M checkpoint. So in G1, it makes sure that the DNA is intact before it decides to replicate. During the G2, it checks that replication occurred properly, that chromosomes were duplicated properly. And then M is making sure that all the spindle fibers are attached properly here. Now, this is an important checkpoint because sometimes mistakes are made where two chromosomes or two sister chromatids, as we'll talk about in a little bit, do not separate properly because the spindles were not attached properly. That will result in certain diseases like Down syndrome or other trisomies, where you have 3 copies of a chromosome in a cell. Most trisomies actually result in miscarriages and things like that, but some do survive. And as I said, Down syndrome would be an example of that. So if there's an issue with the M checkpoint, or there's an issue with some of the proteins or the replicative machinery that exists in the cell, that could result in certain genetic disorders as you get older in life. All right, so there's certain molecules that help to actually control these different checkpoints. They're called cyclin-dependent kinases and cyclin. They're both proteins, okay? So the thing that actually drives the cell cycle is something called the maturation promoting factor, and that is a mixture of both cyclin and a cyclin-dependent kinase. And as you can see here, a cyclin-dependent kinase is actually present in the cell at all times, okay? But it's not activated until a cyclin binds to it. Now, cyclins are only produced at specific times and produced in certain concentrations. So there's different cyclin-dependent kinases that go with different cyclins, which will promote moving on to the next phase of mitosis. If everything works out, the cyclins will accumulate, bind to the cyclin-dependent kinase, and it'll say, okay, we can go through the next checkpoint. If there's something wrong, cyclin won't be produced, and if it's not produced, it's going to halt that cell in that specific checkpoint. And either they're going to fix the issue, Or if the issue is not fixed, the cell is going to actually commit cell suicide or apoptosis. So a quick question, what does a kinase do again? A cyclin-dependent kinase. What is this going to do? It's going to go and phosphorylate other proteins, which go and phosphorylate other proteins, which eventually cause some sort of change. Remember back to when we talked about cellular communication and signal cascades. So a cyclin-dependent kinase is an example of that. So as we said here, here's a quick overview of what I just talked about. Cyclin-dependent kinases are produced all the time, but it's only turned on when cyclins are produced. Okay, and there's different cyclins for each part of the cell cycle. The genes for each cyclin are turned on at different points. They bind to the CDK, which will then go and activate other proteins. Now, how does that process stop? Well, there's a special protein that's called ubiquitin that is present, and ubiquitin will actually bind to cyclin and mark it for destruction. So after it's there for a while, it will be destroyed. So if it's being produced, we're going to continue to have cyclins, and we're going to continue to go through the cell cycle. If it's not produced, then you won't. And we halt that process. So once we move into the next phase, we just want to get rid of those old cyclins. So ubiquitin will be involved in that process. Here's kind of showing you how that works. Here's your cyclin-dependent kinase and cyclin bound to it. Ubiquitin is bound right here, and it marks cyclin for destruction. There are certain drugs that we can actually look to see the effect of messing with these different proteins, and we can use that to study things like cancer. So these drugs would change the shape of either cyclin-dependent kinase or cyclin or the MPF in general, and maybe ubiquitin. And if we stop these things, we can look at the effects on cells to see that, maybe it'll result in cancer, or maybe it'll result in other issues. Usually wouldn't do this. I mean, we could do this in humans, but they also do these types of things in cell cultures. So how do your cells know when they want to heal themselves? Or if you have a cut, how does it know that it needs to go into the cell cycle? Different cells will release these things called growth factors. Growth hormone is an example of this too, but it's a chemical signal, a hormone, a ligand, that'll bind to receptors in the cell surface. What they will actually do is take cells out of G0, remember G0 means they're just doing their thing, and move them actively back into the cell cycle. They also have this thing called contact inhibition. So if your cells are in contact with other cells, they won't want to divide. But as soon as they're cut, or if there's no cells next to them, they're going to say, okay, I need to grow until I'm in contact with other cells. That's kind of how your skin works. That will trigger mitosis. So this is actually showing you a signal cascade. Now, you don't have to memorize this, but you can see a growth factor binding to a receptor. We have phosphorylation cascades, eventually activating different proteins, which will eventually stimulate the cell cycle. Here's showing you contact inhibition. Cells have been removed. It causes them to actively start dividing, and they will be replaced. In cancer cells, though, they don't know how to stop this process, so they just keep dividing over and over and over again. Here's showing you some of the receptors on the outside surface of cells where the cells can recognize if they're in contact with other cells. So if they're not in contact, they're going to stop the process of mitosis from happening. Or excuse me, if they're not in contact, they will start the process of mitosis happening. If they are in contact, they're going to stop it and go back into G0. So in summary, the cell cycle consists of interphase and mitosis. Interphase consists of G1S and G2, as well as G0, which is that complete going out of the cell cycle phase. There are checkpoints that will allow the cell to move from one phase to another. And remember, you have to meet certain criteria. The DNA has to be replicated. It has to grow properly. The mitotic spindle fibers have to be attached properly in order to get from one phase to another. And the last thing is that the cell cycle is controlled by cyclins and cyclin-dependent kinases. And, you can think about that as kind of a cell cycle control where the cyclin-dependent kinase needs the cyclin in order to be activated. It goes and phosphorylates other proteins. And then once cyclin stops being produced, ubiquitin will go in, destroy the cyclin, and then the cell will have moved into the next phase after that point. So once the cell gets the go-ahead, it can undergo the process of mitosis. It must pass all the checkpoints. So that means it has no DNA damage, the DNA was replicated properly, and the cell has grown enough. But before we talk about that stuff, we have to look at chromosomes and understand chromosome structure. The centromere part of the chromosome is the central part right here of a duplicated chromosome, as you can see here. The telomeres, remember, on the outside parts of the chromosome. The centromere is actually the spot in which the spindle fibers, which are going to pull the chromosomes apart, are going to attach. And remember this, human cells contain 46 chromosomes. You get 23 from your mom and 23 from your dad. So each chromosome has a matching partner, which is called a homologous chromosome. It has the same genes on it, but sometimes it has maybe different versions of the gene. So like you have genes for eye color, they'd both be on the same chromosome, but you can have different versions. Maybe you got a brown from your mom and a blue from your dad, for example. But we're going to talk about that more in later chapters. Body cells have 46 chromosomes, and the gametes, sperm and egg, have 23, which is half. Okay, because body cells have 2 copies of every chromosome, they're called diploid, or 2N. So they have 2 copies of every chromosome, one from mom, one from dad. Haploid cells only have 1 copy. So gametes, sperm and egg, are haploid. They only have half. They have the full recipe book, but they only have 1 copy of the recipe book. So that's why it's deemed N as opposed to 2N in diploid cells, because they have 2 copies. Prokaryotes are always haploid. They'll only have 1 copy of their DNA. And at some point, every eukaryote has some sort of haploid stage if it does sexual reproduction. Chromosomes also can exist in 2 states. We've talked about this before. Before they exist as a chromosome that has condensed, they exist as chromatin, which is just a jumbled up mess of DNA in the middle of the nucleus. So they don't actually exist as an individual chromatid except for when they're pulled apart during mitosis. When there's only 1 copy, or when there's only 1 whole copy of all the DNA that exists as chromatin. Then during S phase, DNA is replicated as chromatin, and once it condenses, it then has 2 sister chromatids attached. So each of this, and this is an identical copy to the other one. This is not the same as homologous chromosomes. In homologous chromosomes, there'll be a whole nother chromosome sitting right here next to it that has the same genes on it. More about that when we get into meiosis. So here's showing you chromatin, the jumbled up mess of DNA, and then it gets wound around histone proteins, eventually forming the chromosome structure. And this is again showing a copied chromosome. Each of these is identical to each other, and they're called sister chromatids in that case. So now onto mitosis. Mitosis is the process of cell division in the cell that is a eukaryote. So this requires something that has a nucleus, membrane-bound organelles, etc. The steps of it are prophase, metaphase, anaphase, and telophase, and we're going to go through each one individually, and you're going to want to, in your notes, kind of write down characteristics of each step. So prophase, I have 2 pictures here because they have early prophase and late prophase, or sometimes it's called prometaphase, but this one is particularly showing late prophase. The chromosomes will go from chromatin to chromosomes. So the chromosomes will said to have condensed. What you actually see here too are 2 sister chromatids and 2 homologous chromosomes. So the red X, there's 2 sister chromatids on this red X, and then its homologous pair or partner is right here in blue. 1 from mom, 1 from dad. The homologous pairs don't really matter during mitosis. It's not going to matter until meiosis when we talk about that process, which is going to be in the next lecture. So the chromosomes condense, the nuclear membrane will eventually disappear, and then these things, which are called centrioles, which are special organelles which specifically produce the spindle fibers that will pull the chromosomes apart, and they only really condense and appear during mitosis. So they go to opposite poles, and what's eventually going to happen is the spindles are going to attach to these chromosomes and pull them apart. So that's prophase. Chromosomes condense, nuclear membrane disappears, spindles move to opposite centrioles, and spindle fibers move to opposite poles. During metaphase, which is the next phase, is the chromosomes will line up in the middle. That's how you can remember it, metaphase, middle. So they're all going to line up, and the spindle fibers, notice how are attaching. At this point, they have actually a cell cycle check to make sure that all of these are attached properly. During anaphase, they start to get pulled apart, the sister chromatids. Now you can see one chromatid goes to one side, one chromatid goes to the other. And then during telophase, cytokinesis starts usually. Nuclear membranes form around the original genetic material. And what we have here are individual centrioles and two new cells forming. Here's showing you some real pictures of this stuff. So this is showing anaphase, or excuse me, metaphase. This is prophase, this is anaphase, and then telophase at the end. So the spindle fibers that actually attach to these chromatids form at what's called the kinetochore. Okay, so that's how it attaches at the centromere. The spindle fibers will actually contract and pull the chromosomes apart. So how does this actually happen? So they wanted to know, do the centrioles pull the chromosomes apart from this spot over here, or does it actually like slide along here and destroy so it would be like this is almost like a Pac-Man eating this as it gets pulled apart. You'd think it would be pulling them, but in fact it doesn't actually pull them. And the way they found this out was they actually radioactively labeled, like they pretty much do everything, they radioactively labeled these proteins right here that are in the spindle fibers, the microtubules that make up the spindles. And they destroyed the radioactivity. So in fact, this is not, the spindle fiber still exists, but just this part is not radioactive. If it was pulling from this region right here, you'd expect this mark to move. If it was coming from this region, that mark should stay the same. And what they actually found was the mark did not move. So that shows you that the spindle fibers actually are destroyed at the chromosome. And it's almost like this eats it like Pac-Man as it goes down the spindle fiber. So, in summary, mitosis has 4 phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes condense and the nuclear envelope disappears. In metaphase, they line up in the middle. Anaphase, they start to get pulled apart by the spindle fibers, the sister chromatids do. And then in telophase, the cell starts to do cytokinesis, a new nuclear membrane forms, and the cells can eventually start a new life as their new cells. Here's showing you cytokinesis in real cells. It's showing the pinching process right here. Okay. In plants, though, it does it a little bit differently. It doesn't just pinch. It actually forms a new cell wall right here, and then we'll divide into two. This preliminary cell wall is called a cell plate. So you want to make sure you know that. And vesicles form this new cell plate, and eventually it'll split and divide into two. Because obviously in plants, they're more rigid because they have cell walls. All right, so we're going to finish with this for this lecture. If you have any questions, feel free to email me. I'll be checking my computer relatively regularly.